Chemical digestion. So in this lab, we're going to study enzymes and their role in chemical digestion and the characteristics of enzymes. We all know that digestion starts with mechanical digestion and that transforms food into macromolecules, and then the function of the enzymes is to transform macromolecules into micromolecules which are ready to be absorbed by the um, capillaries of the intestinal villi. So enzymes are proteins made by the body. They are biologic catalysts. What is a catalyst? Catalyst is a product or a anything that increases the rate of chemical reaction. However, it doesn't enter into the final product. It is not transformed. When two products enter or two molecules enter into a reaction, then the final product is the result of the transformation of the two products or three products, whatever was in the reaction. However, the catalyst doesn't transform, it doesn't change, it comes out of the reaction unchanged. So, what does it do? It increases the rate of the chemical reaction, it increases the um, efficacy of the um, chemical reaction. So, it's not consumed in the chemical reactions, it increases here. Okay, it reduces the amount of energy needed to start the reaction because our cells perform lots of chemical reactions. Millions of reactions occur in our cells um, within a minute. So, all these reactions consume a lot of energy. However, enzymes help to reduce the amount of energy needed to start the reaction. However, there is a catch for it. Enzymes are very specific to their substrate. Enzymes act on substrates. Substrates are the molecules on which the enzymes act and on which the enzymes help to speed up the reaction or make the reaction very efficient. So they are very specific. So let's go here into our example. So this is an enzyme. It is called the sucrase. Enzymes end with the A's, three-letter A's. Sucrase means it acts on sucrose. So sucrose is a disaccharide made up of a monosaccharide, fructose, and monosaccharide, glucose. Adding water makes it a hydrolysis reaction. And the water which is found in the intestines together with the sucrose, which is a product of the carbohydrate digestion. It's a disaccharide. However, it cannot be absorbed in the intestinal villi. It needs to be transformed into monosaccharides. Okay, so water plus the disaccharide sucrose in the presence of sucrase is going to be broken down into monosaccharides, glucose and fructose. Now, this protein has a special three-dimensional shape, and the three-dimensional shape is on every part of the protein, and especially it is very specific here at its active site. This is what we call it, the active site. It is very specific. This is made especially for the sucrose. another disaccharide cannot come on this, or even it comes, it doesn't fit on here. Okay, so that's what I mean. It is very specific. Its enzyme is made specifically for its substrate. Now, the other specific thing with enzymes is that they are first, they are highly specific, as I said, then they are very efficient, and then they catalyze the reaction with very specific, very extremely rapid rates, and they don't enter into the reaction itself. They come out of the reaction unchanged. The substrate changes. As we see here, the disaccharide became monosaccharide. However, the enzyme didn't change and is ready now to find another molecule of sucrose and work on it. Okay. And the third thing is that enzymes are under the subject of cellular controls. It means that the cell regulates the production and the concentration of the enzyme needed at a given time. During digestion, more enzymes are produced. Who is controlling that? The cell's genes. Now, you know all about enzymes from your digestive system studies. We know enzymes that work on carbohydrate digestion, protein digestion, fat digestion, nucleic acid digestion. And so these enzymes, they start working. We have the salivary amylase that start in the mouth, and then it is denatured in the stomach because of the acidity of the stomach. And then we have the pancreatic amylase that breaks down the carbohydrates again into disaccharide, lactose, maltose, sucrose. And in the brush border of the intestines, these are broken down into monosaccharides. Lactose becomes galactose and glucose, maltose becomes two molecules of glucose, and sucrose becomes glucose and fructose, as we saw in the example. Proteins are broken down by the first by the stomach acid, and then enzymes trypsin and chymotrypsin break it down into amino acids, smaller peptides, and then brush border cells, they break it down, the smaller peptides break it down into amino acids. Fats, of course, starts with the lingual lipase, then there is the gastric lipase, then part of it is emulsified, and then pancreatic lipases do the final digestion into fatty acid and monoglycerides that are absorbed, enter the intestinal cells, and then part of it becomes larger protein combined with larger molecules combined with protein. They become chylomicrons, and then lacteals absorb it. And smaller chain fatty acids are absorbed in the capillary of the blood in the villi of the small intestines. So these are just a reminder of refresher of the enzymes that are found in the digestive system. So these digestive system enzymes, they are hydrolytic, or what we call it hydrolases, because they need water to work on them. So amylase is the enzyme that breaks down the starches. Then we have the lipases, then we have the proteinases. So all these enzymes, they are proteins, and they work on specific substrates. They are hydrolases. They all are temperature specific. They have their specific temperatures where their function is at its optimal. Because they are proteins, many enzymes are denatured or deactivated by heat, or they slow down or they stop at freezing or lower temperature. They are pH specific. Uh, some have special their optimal pH at which they can work very perfectly. For example, pepsinogen becomes active pepsin at a very high acidity or a low pH. So most of our enzymes in the body, they work at neutral pH 7. Um, so here, let's go into our experiment. So here in the experiment, we're going to um, see the starch. We're going to take the starch and water together with the enzyme amylase, and we're going to see the, if maltose has been produced. If starch is digested by the enzyme amylase, then maltose will be there. If not, then starch will remain as starch. So we'll check for the presence of starch. We're going to check for the presence of maltose, and we'll check if there is a digestion, and if it's digestion, if it's complete digestion or incomplete digestion. Um, so as here we will see in the experiment how if the starch decreases, it means that the starch is being digested into maltose, so the maltose is going to increase. So we have to detect the starch and to detect the maltose, and for that we need special detection methods. They are special chemicals that we'll see, or special reagents. So as we know, starch, when you eat the starch together with the amylase in the saliva and the water which is found in the saliva, will transform starch into maltose. Um, that happens here. This is the whole carbohydrate, lots of glucose, um, and then by amylase, um, it is broken down into maltose. This is another maltose. Maltose is a disaccharide. And then this disaccharide later is broken down into glucose by maltase. So here this is maltose. So to do that, we're going to take test tubes, um, and then, sorry for this, and then we're going to take a 250 ml beaker and the water bath, um, and then spot plates and droppers. As we go into the experiment, I'm going to show you all these. We need a dropper bottle of maltose, of amylase, and starch. These are the reagents that we're going to use. So with every experiment, we need controls. So controls, they provide a known standard against which the comparisons are being made. When we do the test tubes, so at the results, we're going to compare it against the controls. So we have to prepare the controls. The controls are 3 tubes. We're gonna mark them as 1A, 2A, 3A, and then the experimental tube, they are 4A, 5A, 6A. So let me go to the chart and explain here on the chart. So these are the test tubes. Of course, they are made up of glass. And then these are the first three are the controls. These are our controls. And on the first control, we have to control the presence of amylase. Here, this is amylase in here, the purple color. So we add just amylase and water. There is no reaction in here. And then starch and water. Again, we don't expect any reaction. We're just going to check the presence of starch in here. And then this is just maltose and water. So these are our controls for the three reagents that we are using here. Maltose, amylase, and starch. Now in the experimental tubes, we're going to add the fourth one. Is important tube that we add the amylase, and then we denature it by boiling it for 4 minutes. So boiling of the amylase, it's a protein, it will denature it, and then we're gonna add the starch on it. The fifth tube will work at optimal temperature, 37 degree. It's not denatured. And then the sixth tube, again, we have good amylase. However, the temperature is not optimal. We incubate it at freezing temperature, 0 degrees centigrade. So we'll take all these 6 tubes, 1, 2, 3, 4. So this is the same tube, 5, 6, and put them in water bath. The 5 of them, we put them in water bath, and the 6th one, we put it in ice bath, in ice water bath, and incubate it for 1 hour. After 1 hour of incubation, we were going to see if the starch has been hydrolyzed. Remember, I told you digestive enzymes they hydrolyze, okay? Because we added water and we added the enzyme to facilitate the reaction. So they are hydrolyzed. If they are hydrolyzed, it means that the starch has been converted into maltose or partially converted. Okay. And how are we going to do it? We're going to detect the presence of starch or the presence of maltose or the presence of both if there is partial digestion. Okay, so to do that, we need to do 2 experiments. One of them is to check for maltose, the other to check for starch. To check for maltose, we have to do the Benedict test. And for that, we need to, again, to boil the water in the 250 ml beaker. We put 125 milliliter of water and make it and wait until it's boiled. Meanwhile, until this is being heated up to boil, we check for the starch by Lugol's solution. Okay, so we're checking for starch and we're checking for the presence of maltose. So to check for starch, we're going to take a spot plate. These are plastic or glass plates that have tiny little wells in them. Okay, it's called the spot plate. So on the spot plates, we mark it as 1, 2, 3, 4, 5, 6. Okay, and we're going to put 3 drops from all the tubes that we took, the incubated tubes, into from 1A tube, we put it in the well 1A. So here from the tube 1A, we put 3 drops of it here, 3 drops of 2A in here, 3 drops of 4A. And then we're going to, here, let me go here, we're going to add 2 or 3 drops of the glucose solution. Glucose solution is called the IKI solution. It has iodine in it, and it tests for the presence of starch. If there is a starch, then it turns into very, very dark blue, blackish, kind of very dark purple color. Okay. Because the color of glucose solution is a kind of light brown solution. So if there is a starch, then it's going to turn into very dark color. If there is no starch, this is its solution. That's the color of the glucose iodine. Okay, so positive test, starch is present. Negative test, so when is the starch negative? Starch is negative either if we didn't add any starch in the control, or in the test tube it's negative if the starch has been digested into maltose in the presence of amylase. So then, according to the results, we put in this table, we put our results. So here we put in number well, number 1 well, number 2 and 3, 4, 5, 6. So number 1, what was in number 1? Number 1 had amylase and water. Is there any starch in it? No. So do we expect any starch in there, any color change? No. No color change. So Lugol is negative. Here we added starch. So what do we expect in here? We expect color change, yes. And then Lugol is positive. Let's check. This was negative. And here we have color change, positive. Number 3 is negative. What did we add in number 3? We had maltose in here. Is there any iodine? Do we expect any positive? No. No color change. Now let's go into number 4. What did we add in number 4? We added a starch, we added amylase, and we added starch. However, the amylase was denatured. So do we think the starch is going to be digested by a denatured amylase? Probably not. Probably not. Sometimes when we don't give enough time for the deactivation, sometimes there is a little bit of digestion, but usually not. So now let's see here, number 4, there is color change. It's not as this one, but there is some partial color change. It means that the starch has been partially digested in there, or maybe 75% digested. So is there color change? Yes, it is positive. Now let's go back here. Number 5, it's closer, as if it's closer to here, to number 1, to our control. Remember what were controls? They were the standards, known standards against which we are comparing it. We know that this is the negative and we know that this is the positive. So we're comparing against those. Okay, so here this is very similar to this. So it means that there is start digestion and there is no color change. Almost there is no color change. So starch is negative. No color change. And it's negative. Now let's go into our incubated on ice tube. Here, a starch, there is a starch in here. There is color change in here. There is starch. It means that there is no digestion, or maybe just a little bit digestion. Maybe we should have given it more time because it was on freezing temperature. So we write color change. Yes, it is starch positive. Okay, so now we know these are the tubes, and where was the starch digested and where was the starch that was not digested. Here starch, these are our controls, and here our test tubes. Here is the starch has not been digested or maybe partially digested. Here it's completely digested, and here again it's partially digested. Okay, these are pictures, ideal pictures with, this is the glucose solution, and this is the starch solution that we use. And when you add glucose solution into starch, this is the color change, if it's complete detection of starch. Okay, so here we checked about the starch. Now let's check about Benedict's. Benedict reaction is to check for the presence of maltose. So if there is digestion of the starch, then a starch is negative and maltose is positive, because the result of digestion of starch is maltose. So we have to check for the presence of maltose, and that is by using another reagent called Benedict's reagent. So Benedict's reagent is, we add, remember our test tubes. Okay, so we took 3 drops of these test tubes and put it on the spots for iodine. We put it here to check for the starch. The rest of the tubes, we will put it in the boiling water. And of course we're going to add 7 drops of Benedict solution and then place them in boiling water for 5 minutes. Here is what we did. This is in our lab. We took the beaker, 125 ml of water, and then we put all these 6 tubes in boiling water for 5 minutes. So all of those, they do have Benedict solution in it, 7 drops. So if there is maltose, then the color of the solution is going to change according to the concentration of maltose. So here again, this is a picture from our lab, and this is tube number 1, 2, 3, 4, 5, 6. Usually our tubes, here 1A, 2A, 3A, 4A, 5A, 6A. So the first tube, if you remember, what was in it? It had amylase in it. Is there any chance that there is maltose in it? No, unless there is contamination, which happens sometimes. But here, there is no maltose in it, and the color of the dropper of the solution at Benedict's, its normal color is blue. So here, see how it is changed, stays blue. You put 7 drops of Benedict into the amylase here, and it stays blue because there is no maltose. This is our starch control. So when we added some Benedict, it turned a little bit bluish green because probably the tubes were dirty from a previous lab. Sometimes it gets contaminated with maltose. That's why there is a little bit of color change. This is our control for maltose. Remember, tube number 3, we added maltose, right? Tube number 3, we added maltose. So we expect when we put Benedict solution, we expect a lot of color change. When there is a big concentration of maltose, then it turns into red. Tube number 4, we added amylase and starch, but the amylase was denatured. So do we expect any reaction, any digestion of starch? Amylase is not working. So if it doesn't work, then there won't be any maltose, so it will be negative, like the control. Now tube number 5 had amylase and starch incubated at 37 degrees centigrade, and then we added the Benedict solution and see the color change. And here, tube number 6, which was frozen, which was incubated on ice water, again there is reaction, again there is some digestion. This is done with one group, and this is done with another group, which had similar results. Okay, so the results of our experiment in here. Now let's go back. So here, Benedict test, was there any color change? No. Is it positive or negative? It's negative. Now the second one, there was a little bit of color change, little color change, little color change. I can say positive, negative maybe in our case of the experiment. Here, this was what there was color change. Yes, it became red, and it is positive. Here, this was denatured, so the starch didn't get digested, so we didn't get any maltose, so the Benedict test was negative and there was no color change. In the fifth tube, there was color change, and here there was color change a little bit, and so positive and positive. Okay, so as you see here, I also added all the results in here. Um, Theoretically, you should have done this on and at home and sent me all the information as an assignment, but I wanted to make it easy for you. But what I want you to know what's happening, and I will quiz you on that on May 18th, next Monday. So I want you to understand exactly what's happening with the experiment and how we detect the presence of maltose and the presence of starch. We detected the presence of maltose by Benedict solution and the presence of starch by Lugol's solution. If there is starch, it means there is no digestion. If there is maltose, it means that there is digestion of the starch. So here again some pictures for you here. This is the regular starch solution, and this is the Benedict solution. If it's negative, if we add Benedict solution on the starch, this is what we're gonna get. It will turn into its regular blue color, no color change. If there is color change from yellow to orange to from green to yellow to orange to red, it depends on the concentration of maltose. Red means there is high concentration of maltose by percentage. Okay, so here is again 0%. This means 0.5%. This means there is 1.5% maltose. There is 2% maltose or higher. So as digestion occurs, sometimes if we give more time, so the starch, as the starch decreases, the sugar will increase and the digestion will occur more, and according to that the color is going to change. So that's why sometimes we see different colors as we saw here. Let me go back. As we saw here, probably if we have given more time, this would have been more red. Sometimes there is partial digestion in experiments. Sometimes we do have contamination from using, probably some students here, they used the same pipetter that had maltose in it to put here the amylase or the Benedict solution. Probably there was some contamination, that's why we got some partial digestion. So that's why we do have this color indicators, and that's why we have the control to compare against a known standard. Okay. So, and this is our interpretations that I explained. So when amylase is denatured, the starch will not be digested. And that was in tube number 4. And that's why starch was not digested Tube number 4 tested positive for starch and negative for maltose. Okay, so one of the questions, for example, I might ask, which tube tests positive for maltose? Of course, the red one. Which tube contained, in this case, which tube contained the heat-treated amylase? If it was heat-treated amylase, means that it was negative for Benedict solution. So tube number B. Okay, so what I want you to know from this lab is um, that we check for reagents, we check for the products of an experiment by adding reagents. And these reagents, they change color according to the presence of the product. In this case, with the product that we are looking for, the presence of maltose. If maltose is present, it means the starch was digested. It means that the amylase is working. If maltose is not present, it means that the starch is there and the starch was not digested. If the starch is not digested, either the amylase is not working or the temperature is not optimal. So that's why we do the experiments to see and to check. And of course, we always need the controls to check against the known standards. All right, so there are other experiments too that to check for proteins and to check for the lipids. But here, this year, we're going to do just the experiment which pertains to the digestion of carbohydrates. Okay.